All right, in the last video, we talked a little bit about the life cycle of a somatic cell, and somatic cells can undergo a type of division known as mitosis. So mitosis occurs in small phases, and they are here: prophase, metaphase, anaphase, and telophase. Now let's take a look at each of these phases separately. So what happens during prophase, which is the first phase? Well, our chromosomes condense. And so if you were looking at this under a microscope, you can finally see our chromosomes, right? They've gone from chromatin and they've coiled up and they've been copied, and the two sister chromatids were put together, and now we have that little X-shaped chromosome. The nuclear membrane, the nucleus, starts to break down, and these crazy little microtubule fibers start to form, and these are known as spindle fibers. And these spindle fibers are going to be the protein machinery that push and pull these chromosomes around in the cell. And then on the outside, right, you're going to have these little organelles known as centrosomes, and a centrosome is made of a pair of centrioles, and these are kind of like winches or like a rod and reel. They're connected to these spindle fibers, and they're going to kind of like crank them in or crank them out to allow the cell to divide. Now, what follows prophase? That would be metaphase. And I think this is the easiest of the phases to recognize. I think metaphase is the middle phase, right? And so what happens during metaphase is our chromosomes get pulled and pushed to the equator, right? So this is like the equatorial plane, right? Right across the center. And I mean, it makes me think of like picking teams for like a kickball game, right? Everyone lines up in the middle and you start pulling, okay, that chromosome will go there, that chromosome will go there, right? So you get ready to divide it. I wouldn't worry too much about pro-metaphase, right? Pro-metaphase would obviously just be like kind of in between prophase and metaphase, with the spindle fibers attached to the centromere, that little protein that holds the chromosomes together. But really, at the end of the day, metaphase is all about getting these chromosomes lined up equally along the middle. Metaphase, middle phase. That's what I always tell myself. Now, after they're lined up in the middle, they need to get pulled apart, right? And they need to be separated. And this is anaphase. So I think anaphase is the away phase. And what happens here is the centromere splits. That little protein that was holding those two sister chromatids together to form that single large chromosome is broken. And so now we have two of those sister chromatids moving in opposite directions. Now remember, these guys were attached together and they were sister chromatids, so they were identical, right? And so maybe this is, if we just think about, maybe this was the type 1 chromosome, this was the type 2, this was the type 3, and the type 4. These guys were identical, they were identical, they were identical. So it makes sense to split them apart, because now these chromatids will slowly make their way to the opposite side of the cell and ensure that the two new daughter cells will each have the right number and right types of chromosomes. Now, sometimes this division of chromosomes does not work out right, and we'll talk about what happens when that does happen. Now, following anaphase, it's time to start putting the pieces back together, and this is known as telo or telophase. During telophase, I think it's like the tearing phase, because it looks like the cell is getting ready to tear apart. What happens during telophase is those sister chromatids, or those lone chromosomes, start to unwind back into chromatin. The nucleus starts to reform. The spindle fibers and the centrosomes start to break down. And in the cytoplasm and cell membrane are getting ready to divide. Now, telophase or telophase would be the last stage of mitosis, because remember, mitosis is all about dividing the nucleus or dividing the contents of the nucleus. And since our nucleus is reforming, well, that would be the final phase of dividing the nucleus. Is now we're going to have two separate nuclei. And that takes us to our final separate stage of the somatic life cycle, and that's cytokinesis. During cytokinesis, the cell membrane and the cytoplasm finally divides and separates. But there's something important to note here, that cytokinesis is not a part of mitosis. It's interphase, mitosis, then cytokinesis. Cytokinesis happens after telophase or after mitosis. Now, if cytokinesis failed to occur, what you would have would be a single cell with two nuclei, right? And if they're both the command center, you could see how complicated that could be, the cell receiving two different commands or two of the same commands at the same time. Now, let's look at all of it together, okay? So I'll put my face right here in the middle. So interphase, right, is where the cell is busy growing and making copies of the DNA and then getting ready to divide. Remember, that's 80 to 90% of the cell's life is an interphase. Now, once it's ready and it's made copies of its chromosomes, it can then enter mitosis, right? That would be the division of the nucleus, right? Making sure to divide the chromosomes equally. And so we would have our prophase where we can finally see the chromosomes and we get them lined up in the middle during metaphase. And then we start to tear them apart in anaphase, right? Tearing that chromosome apart and sending those sister chromatids in opposite directions towards the poles of the cell. And then eventually the nucleus would start to reform inside of both of those daughter cells, right? And this would be telophase or telophase. And then mitosis is over. And then the last and final step would be fully separating the cell membrane and the cytoplasm in cytokinesis.